to see that our company is designing methods based on calculations of the relations of genetic sequences. Designing methods based on calculations of the 有基因序列的亲属设计有感疫苗探头。Now, this research we are trying to use the science mathematical methods, trying to find out the immunity point of the unknown flu viruses. We will compare it with the known viruses, find out which different part is it, and different part might be the immunity point. And in here we will compare about the PV2 sequences, HA sequences, and NA sequences. The reason that we want to compare the HA sequences is that in this movie you see that HA is like a key, connected to the surface of the cell to let the viruses get it into the cell and produce it. And after producing a lot, NA it will come out of the cell, and getting out of it. To get out of this, they need to have NA, like a scissor, to cut it off, like here. You see, like the NA is cutting off this, so the new virus can get out of the cell and go and infect other cells. And the PV2 is the evolutions of the influenza A viruses of each type.
In our research, we propose mathematical and bioinformatics methods trying to locate the vaccine immunity point. And in our research, we use two different kinds of mathematical methods. One is UPDNA, and one different method using arithmetic average. And another one is MLE, maximal likelihood estimate. UPDNA is to using a similar metric try to construct the phylogenetic tree diagram. And through explaining, we gradually combine two most close biological groups into a high-level group. And in our research, we use UPDNA to calculate because it is the most frequently used phylogenetic analysis rate. And here's MLE. This one is a likelihood function. In the function, x1 to xn means the 14 influenza A virus sequences we find out from the NCBI and the database. The theta is the sequence we need to compare with. And in our research, we compare with PV2, HA, and NA. And after we calculate the possibility max function, we can find out the relationship between 14 influenza. In case maximal likelihood estimator of biological sequences. And here we use two-side ratio approach to try to find out the best estimation of theta. And after we calculate with these two different kinds of mathematical methods, we can come out with the routine tree and fuller tree, like this tree.
And we find out the 14 viruses on NCBI database, National Center for Biotechnology Information, and we calculate its PV2, HA, and NA. And in this routine tree, it's calculated by the V2. As the picture shows, you can see that H5N3 and H7N3 has a close relationship. Also, H7N7, H7N9 has a close relationship. These two pictures is before all sequence comparison. Those two pictures is all sequence after all sequence comparison. Those two pictures is using maximal likelihood estimator by deleting the head and tail. As you can see, H7N1 and H7N9 also has a close relationship in the same as the routine tree. But after all sequence comparison, we found the H7 group get closely together. It means that it has a more closer relationship in this routine tree. And in this maximal likelihood estimator, we have, if we have to get more accuracy, we have to cut the head and cut the tail, and just get the middle part, because the length of the 14 viruses are not the same. Some are short and some are long. So take H5 for example. The star means that the three sequences are the same. The one without the dot means it might be the brand or just different. It may come out with the 17, 20, 23, and so on. 17, 20, 23, and so on. And in total, we find out the 400 different locations. In traditional inventing a new vaccine, scientists have to do all the sequence by these 1,849 sequences. And now, using our mathematical method, we just have to do the 400 different locations, same in the example. Okay, now talking about is about HA analysis. I'm now going to talk about the HNA analysis. And here you can see that these two are the before all sequence comparisons, and these two are the after all sequence comparisons. And this two is the maximum likelihood tree diagram of the 14 viruses after all sequence comparison, according to head and account. And you see first see here that in this before all sequence comparison, H7N9, H10N7, and 7 and A sequences has a really close relationship. Same in here. H7N3 and H5N3 and A sequences has a close relationship too. And but after doing the after all sequence comparison, we found that it didn't have a lot of difference between these two pictures. So it means that this group has a very close relationship, and this group of viruses has a really close relationship too. And moreover, we do the maximum likelihood to get a secondary analysis and to get more accurate experiments. So we find out that the H7 groups here has a really close relationship, and here H7N3 and 5N3 has an A sequences has a really close relationship too. So we do the same methods with the HA. We compare the differences between it. The star means that they have the same parts, and without a star, that means that they have then were just different. So we count out that this H5N3 and H7N3 they have 191 different locations between them two, and the total PP is the 1,410. So we need to just find out this 1,901 different locations. And the same in this N7 group, H7N7, H7N9, H10N7. You can see that there are 623 different locations, and the whole PP is 1,438. So we just need to do this 623 different locations to find a unity point. So this mathematical methods, you see that it's we can save a lot of time and money. So while developing a method with biomedical information research, a highly inconsistent ratio is frequently encountered, namely the best locations of immunity. So in this research, we are the first one to conduct computer analysis on unknown viruses, and then compare with sequences of known viruses that is through in1's dynamic programming algorithm. So without seeing this mathematical methods, you can see that this picture is the conventional R&D process of inventing a new vaccine. The scientists will first foster new influenza viruses and run a 2D electrophoresis. And they will keep on this step on and on to find out the right part and the immunity points. And the immunity points can come up with a new vaccines. So then we will do the secondary analysis to help the doctor to help the patients to against the new influenza viruses. So in order to save a lot of time, we added science mathematical methods in here to save time. We use the reverse genetic procedures in here. You can see that this is a non-influenza virus, and this is a standard virus. We will first choose the non-viruses NA and HA sequences and combine with the standard viruses other 6 parts without HA and NA, the other 6 parts, and we will combine it into a new influenza A viruses, and we can use it to invent the new vaccines to save a lot of time. And then we think that this kind of mathematical science methods really help humans a lot. Just briefly saying that we just need to get the influenza A virus sequences of the PB2, HA, and NA on the NCBI database, the national centers, and then compare it with the unknown viruses sequences, and compare just the difference. And the different part might be the immunity points for scientists to invent the new vaccines. So we think that this kind of mathematical methods can save a lot of time, about 3 months to 6 months. So yeah, this is the end of our speech. Thank you for listening.